To them again. If you have questions here in the room, please come up to the microphones. But let's go ahead and start with a question online.
All right, this question is for Andrew. Applicable CGMP and post-marketing reporting, are they applicable to Class 1 devices too? Does the addition of a drug raise the manufacturer's reporting requirements for the device component?
So going into one of the initial points of my talk is that combination products, by virtue of combining the two medical products, create a unique type of product. And so by virtue of them being a unique type of product, there's unique regulatory considerations and regulatory requirements that are expected of them. And so to the extent that it does meet a definition of a combination product, it would therefore be subject to the drug device CGMPs that are outlined in 21 CFR Part 4.
Okay. Microphone number 2, go ahead and step right up nice and close for me, please.
Sure. So I have a clarification question for our first two speakers. So if human factors studies were to observe failures resulting from the content of an RLD's instructions for use, to be eligible for ANDA submission, does the agency expect full alignment with the RLD's IFU, or is it acceptable to make changes to the IFU to address those failures?
That's a really good question. Going to what, ANDAs rely on the previous finding that the RLD is safe and effective. And so the comparison needs to be one-to-one for that RLD. We would expect for them to have the same clinical effect and safety profile. So to the extent there are use errors related to the RLD itself, the comparison would need to be between the two products, and not we would otherwise not expect or allow there to be a difference in that finding.
So just to follow up on that, you're saying if the RLD sees that error as well, then it's acceptable to be able to make the change to allow alignment?
It would be a comparison. So when we look at the comparative use human factor studies, the statistical analysis that's associated with it is a comparison of the use-related use error rates. And so long as you can demonstrate a finding that the use error rate is no worse than the RLD with respect to a certain critical task that's associated with an external critical design attribute, it would otherwise be acceptable.
Thank you.
Andrew, you also can't change the labeling of the RLD, of the generic product to change that indication.
That's correct.
All right, let's go to our next online question, please.
All right, this question is for Kim. Do I have to conduct a comparative human factors study if the user interface of my test products has minor differences from that of the RLD? What if they are identified as other than minor?
That's a microphone there. Hello? Yeah, okay, thank you. That's a good question. I think hopefully my talk, I pointed out that if there are minor differences that we can justify that don't affect the critical design attributes of that user interface, that those would not be anticipated to require any kind of additional information. And one type of additional information that could be included would be a comparative use human factor study. So in that case, the first part of the answer is no. The second part of that question is if there are other than minor differences, one of the things that you need to do with the other than minor differences is really look at what your device combination product is doing in comparison to the RLD and figure out what the best type of information or data that you can provide would be for the agency to be able to look more closely at that potential difference. So when we look at those, a comparative use human factor study might be one of the types of information or data that could be used. Okay. However, let me add.
Sorry.
I had one more thought there. So if you think that is something that you need to do, again, we would recommend that you proactively reach out to us prior to that conduct of any kind of a comparative use human factor study so that you can talk throughout all the different groups within the agency who would need to be involved to help you through that process.
Okay, thank you. Microphone number one, nice and close for me, please.
My question is regarding the development of abuse deterrence product. Most of, not, I should not say most, all of the ADT technologies are patented. And generic companies, they have to come with a novel technology to develop ADT product. At the same time, we have guidances regarding solid oral products on size and shape and trade dress of the product. So my question is, what is FDA's uh, opinion on the criticality or the significance of size and shape guidance over the abuse deterrence bioequivalent product?
Um, in general, the abuse deterrent formulation, any other guidance that, for example, solid oral product, the shape and size that you mentioned, I think that is still applicable. So if that is the case, then you should still follow the other guidance about the requirement. For the abuse deterrent formulation, because it's a solid oral drug product, so it's permitted that the generic version use different excipient, even at a different amount. And quite frankly, sometimes we see that the process itself can impact the properties. So I think that even more than what you asked, but the short answer is the existing guidance still apply, and the abuse deterrent formulation is something rather unique that we need to evaluate from these properties, rather unique properties.
Just adding on that. So in our presentation, we disclosed that. So you know you're a generic cop, so you're not required to use the same abuse deterrent technology, formulations, and excipients, so on and so forth. So you will know your product the best, that you will come up with the most effective manipulation method to demonstrate to us is comparable or no less abuse deterrent compared to the RLD product. So both guidance should be followed. And then with your product specific knowledge to demonstrate that its comparison to the RLD.
Just a follow-up question. So if there is a difference, can it be discussed during the pre-NDA or pre-development meeting?
Add on that, as we were mentioned or describing in the presentation, the evaluation is mostly based on performance. So regardless of the design, the idea is the evaluation should reveal any unique properties for both the reference product and the generic product. The expectation is generic needs to be no worse than the ILD in any of those potential route of abuse.
Okay, let's go to our next online question, please.
All right, this question is for Dajan. Are there any in vitro methods, such as dissolution testing, particle size distribution, drug content, that may be used to substitute for in vivo studies to assess abuse deterrence in the nasal and oral routes?
The short answer to that question is no. So that's what we have to rely on, either oral or nasal PK study to demonstrate the absorption or no less abuse deterrent compared to the RLD. But with that in mind also, I would like to point out that ORS is leading several research projects trying to find potential alternative or substitutions that we can rely on in vitro methods to substitute this in vivo study. But at this stage, it's not mature yet.
Okay, if there's any other questions in the room, come on up to the microphone, but I'll take the next online question, please.
This is for Xiaoming. Can you further elaborate on the strengths or number of dosage requirements, particularly for in vivo tests? In vitro tests, yes.
I think as Daju mentioned in the slide about multiple strengths for both in vitro and in vivo, for the in vitro part, as he was mentioning in his talk, that expectation is if the generic copy propose the strengths that cover all strengths that's available for the reference product, then the expectation is you conduct studies for all strengths. However, if you can establish the composition proportionality, you may also choose to justify potentially to use a bracketing design to reduce some of the testings. But for in vivo, I think he already mentioned that commonly it's intermediate strengths. And there's another about dose or dosage. In the guidance, it mentioned that for the testings, to allow a statistical evaluation, general expectation is 6 units for in vitro studies. That is for the dosage.
Okay, thank you. Let's go ahead and take just one more online question, please.
Okay, I've got a question here for Kim. Let's see. If the RLD does not include an information for use in their labeling, can the applicant include an IFU and identify this as a minor difference, or would this need to be supported by a comparative use human factor study?
Okay, so it sounds like there's a couple of different things buried into that one question. So the first part of it is if there's, I cannot think of an example off the top of my head where a reference listed drug for a drug device combination product does not have an instruction for use of some sort. So that is that would be one question is whether or not the person was actually talking for a drug device combination product or not. Again, when we're talking about comparative use, comparative analyses, we're really looking at these products where there's a drug device combination in place. So that would be sort of the first discernment that we would have to get from that question. I know the person online can't really respond. So, but then the second portion is if there were, is if they could submit something in addition to what the reference listed drug product had. And in general, I will say that usually the labeling, we have a requirement that the labeling has to be the same as the RLD. So therefore, whatever the reference listed drug product has put forward in the labeling has already been determined to be appropriate for that product as it refers to the safety and efficacy of that product. And so therefore, additions to that that fell outside of those rare instances when we can have differences in labeling would not be required or would not be acceptable. And I'm going to look at Andrew and see if he needs to add anything.
So sometimes when you end up having like a pre-filled syringe, there's not those step-by-step sort of instructions because it's an intuitive sort of an administration of the drug product, especially when used in the context of a healthcare practitioner. If there was a necessity to actually introduce a step-by-step instruction, my first question would be, what are the differences between these two pre-filled syringes that would necessitate additional description on how to actually use the product? And so it would go back to what Kim was talking, or Dr. Wisman was talking about in her presentation, going back to the heart of the comparative analyses. Differences in labeling, difference in critical tasks. Does a difference in critical task actually necessitate additional description on the use of the product? And it would need to go through that analysis.
Before we wrap it up, I just wanted to check with the panel based on the questions we had and some of the thoughts going around. Any closing thoughts before we wrap it up? Andrew?
Talk to us early.
Often.
Early and often. There's the wise advice. Please help me thank our panel for coming and talking to us.